You're welcome. In this video, we are considering an overview of cholinergic pharmacology. And that is the rest and digest, because it has got to do with the parasympathetic outflow. This is also called the craniosacral outflow. So yes, considering an overview of cholinergic. Why? Because the parasympathetic nervous system releases acetylcholine, and that is going to cause a rest and digest, and you're going to understand that. And I'm saying a friend of the parasympathetic outflow, and one of the craniosacral outflow. Okay, I am saying cholinergic pharmacology because we are going to study with the drugs concerned with acetylcholine. So cholinergic pharmacology, drugs pertaining to acetylcholine, because here for parasympathetic, it releases acetylcholine. For the both pre and post, they all release acetylcholine. So you're going to look at drugs that can work on acetylcholine, choline, okay, acetylcholine, and all that, and those that to counter the action of acetylcholine on the receptors. Right. So now here, let's go back. Just like the parasympathetic, sorry, the sympathetic. This is the parasympathetic. So we've got here, look at this structure here. This is called the limbic system. Okay, this is the hypothalamus. And hypothalamus, the first part is involved for the parasympathetic, and I essence for the sympathetic activity. We've got some cranial, cervical, this part is the thoracic, okay, and this part is the lumbar. This part is the sacrum. So we have cell bodies within, we have cell bodies alongside. Okay. Now Makacho say, if a sympathetic, yeah, think of a dog chasing you. What are the things that should go on the body? If it is cholinergic, think of now the dog not chasing where we, what about inside, what about them? You understand? What are the changes that should go in again? Do you understand? Right. So let's look at something here. How the para is activated. It starts. The neurophysiology. This is your limbic system. Okay, your limbic system. Now, my limbic system, from your ear, from your temporal lobe, when you realize that, oh, there is nothing serious around me, nothing dangerous around me, I didn't be any mention, then it will send signal to the limbic system again. Or the needs to hear around, no, anything dangerous, best send info to the limbic system. For you know, no call quiet place be, and that's you move be. Then when you din, be at we din, papa friend will die. In who am one, in the honor, so all the signals will come to the limbic system, telling the person say, oh, there is no fear around you. And when that happens, it will send a signal to the parasympathetic part of the hypothalamus. And that will also send a signal through the polysynaptic neuron and come downward. You understand? And this polysynaptic neuron coming from the parasympathetic, they are only going to stimulate cell bodies at the cranial level. This part, the cranial level, in the brain, somewhere in the brain, okay, and then in the sacral area. So they stimulate only cell bodies in the sacral area and the cranial area. So that is why, and then they will send that kind of activity into the part of the body. You understand? That is why when you talk of parasympathetic outflow, we also call them the craniosacral, because the cranial and sacral part are only activated. The cell bodies there are only activated. But when you talk of sympathetic, they are called thoracolumbar because the thoracic parts and the lumbar part are also are activated only. Unlike para, where the cranial parts and then sacral parts, okay, where their cell bodies are activated. Now, when that happens, the neurons over there will be activated, and they're going to send a signal. Okay, so this neuron is coming before this ganglion. This is a ganglion. So you have a neuron coming before the ganglion, and you have a neuron going after the ganglion. And then since it's before the ganglion, this becomes a preganglionic neuron. Okay, these neurons come before the ganglion becomes what? A pre-ganglionic neuron. And the neuron coming after the ganglion will become a post-ganglionic neuron. Now, the pre-ganglionic neuron releases acetylcholine to stimulate the post-ganglionic neuron in the ganglion. And the post-ganglionic neuron also releases acetylcholine. And acetylcholine binds certain receptors, and they are called cholinergic receptors. Simple as that. And you should note that here, for the parasympathetic, their preganglionic neurons are way longer than the postganglionic neuron. Unlike sympathetic, where they have a shorter preganglionic neuron and they have a longer postganglionic neuron. Here, for para, they have a longer preganglionic neuron and a shorter postganglionic neuron. In the same way, for preganglionic neuron of parasympathetic, they release acetylcholine. Post also release acetylcholine. But for sympathetic, the pre-release acetylcholine all right, but the post they release norepinephrine. So you must be knowing that in both parasympathetic and sympathetic outflow, they all release, for their preganglionic neuron, they all release acetylcholine. When it comes to the post, for para, they release acetylcholine. But for sympa, they release norepinephrine or epinephrine. And you know why they are called craniosacral, because they stimulate only the neurons in the cranial part and the sacral part. Unlike sympathetic, which they stimulate the thorax and the lumbar part, but for para, cranial and sacral. So here of parasympathetic outflow is something as craniosacral outflow. Right. Now acetylcholine, because here we know that acetylcholine is a neurotransmitter. Now for the same part, we knew how it was able to synthesize. So we need to know how acetylcholine as a neurotransmitter here, we need to know how we get the acetylcholine. Okay, video. So acetylcholine synthesis. So here at the membrane, level of the membrane, we have what we call sodium choline transporter, because it takes in choline and brings out sodium. So it's a sodium choline transporter. Are you okay? Now this sodium choline transporter will pick choline and send it into the cell. When it goes inside, now on the mitochondrion, it will release what I call acetyl-CoA. Acetyl-CoA. So choline gets inside with the help of a transporter called sodium choline transporter. It picks choline into the cell. And when choline gets in there, mitochondria will bring in acetyl-CoA. And then you have an enzyme that will combine acetyl-CoA is not acetylcholine. So we have an enzyme that are going to combine acetyl-CoA and choline to get acetylcholine. And this enzyme that will combine choline and acetyl-CoA to get acetylcholine is called choline acetyltransferase. They will work on these two and convert them into acetylcholine. Now, acetylcholine will be picked into the vesicle because vesicles move to membranes and bind and release the contents. So it needs to be in a vesicle. So there's a transporter on the vesicle called acetylcholine hydrogen transporter. They send in acetylcholine and they bring out hydrogen ion. And when they come here, acetylcholine comes here, it is in together as ATP. And we have a protein, just like the other one, a protein here and a protein there. This protein on the vesicle is called synaptobrevin, and this protein here is called synaptexin. Okay, so when calcium comes into the cell, it moves the synaptobrevin to fuse with the synaptexin, thereby increasing the fusion between the vesicle and the membrane. And when that happens, the vesicle can really come to the membrane and release its contents, acetylcholine, to bind to the receptor. That's the choline receptors. Are you okay? Or the cholinergic receptors. Now, when calcium doesn't come inside, this synaptobrevin and this cannot fuse. And when they cannot fuse, vesicle cannot fuse its membrane, and therefore acetylcholine cannot be released. But it is very dangerous thing, like when you block, you're going to block the calcium channel. Because if you talk of blocking the calcium channel, all the calcium channels in the neuron, the brain, everything, they're the same. So if you block the calcium channel, you're going to shut down the nervous system because signals, impulses, pain, everything, active potential, they go by working with calcium. So you don't block the calcium channel, but if this part and this part are eating up, that's fine. So let's go over it again. Now this is a sodium choline transporter. To get acetylcholine, this will pick choline and come to the cell. When choline comes inside, mitochondria will bring acetyl-CoA. Acetyl-CoA is not acetylcholine. So there will be an enzyme that will work on acetyl-CoA and choline and convert them into acetylcholine. Acetylcholine will be transported by the acetylcholine hydrogen ion transporter to pick into the vesicle, and it is there with ATP. Now, when more calcium comes inside, the synaptobevrin and textin are going to fuse together. So by fusing, it brings the vesicle close to the membrane. And when they fuse, they are going to release the contents as acetylcholine. So that is how acetylcholine is synthesized. This is an enzyme called acetylcholinesterase. This enzyme is like a scissors. They break down acetylcholine into acetates and choline. Choline is picked back, and acetate goes into systemic circulation. Now let's know some of the drugs acting on the acetylcholine synthesis. So the first drug to know is lactotoxin. Lactotoxin. It is produced by the black widow spider. So black widow spider actually releases what I call lactotoxin. Why? Because they are called black widow spider because this spider, the female ones kill the male ones after sex. They kill them. That's why they are called the black widow spider. They release lactotoxin. So they're going to increase the interaction between synaptobevrin and textin. So here, this lactotoxin is going to increase the binding, okay, the fusion of synaptobevrin and textin. And by way of fusing, they are bringing acetylcholine close to membrane, and therefore it's fused and then bring the content out. And that the one which facilitates the fusion is called lactotoxin, okay, and that is by the black widow spider. And therefore, when that happens, since it increases the fusion, there will be more fusion of the vesicle to membrane, and there will be more acetylcholine. Hemicholinum blocks the choline transporter. Hemicholinum, here, it blocks this transporter and does not allow choline to be picked into the vesicle. Therefore, I'll have no acetylcholine forming. So hemicholinum blocks this transporter here and not allow choline to go inside. Therefore, no acetylcholine. You have vesimicol. It blocks acetylcholine hydrogen transporter. So vesimicol acts here. It blocks this transporter, vesimicol. It blocks the acetylcholine hydrogen transporter and prevents acetylcholine from going into vesicle. And when there is no acetylcholine to vesicle, of course, the membrane might fuse, the vesicle might fuse the membrane, but releases nothing. Why? Because I don't have acetylcholine. Why? Because vesimicol has blocked this kind of transporter, preventing acetylcholine from going into that. So that's how the drug also functions. We have botulinum toxin. It breaks down protease, start digesting synaptobevrin and textin, and then decrease in acetylcholine. So we have a drug called botulinum toxin. Botulinum toxin eats up the protein. This is a protein. Protein here. This botulinum toxin eats up the protein here, So there will be no fusion of vesicle to membrane. And therefore, there will be no acetylcholine produced. So these are the drugs that act on the acetylcholine synthesis. All right. Now let's go to cholinergic receptors. Acetylcholine bind to receptors. Which receptors do acetylcholine bind? We have a receptor called nicotinic receptor. And we have muscarinic receptors. So these two receptors, acetylcholine as a whole, can bind to any of them. Now, why are they called nicotinic and why are they called muscarinic? It is called nicotinic because there was a compound, or let me say a chemical which was detected first. Let me say nicotine, which was taken to the body and realized they could bind to certain receptors. So they named that receptor after the nicotine. That's why you call that receptor in the nicotinic receptor. There is something called the muscarine. This muscarine was taken in, and we got to know that they are binding certain receptors. So we label that receptor after the muscarine, and we call them muscarinic. So we have nicotine binds to its channel called nicotinic receptor. Muscarine binds to its channel called muscarinic receptor. Later on, we found out that acetylcholine can also bind to these receptors, both of them, nicotinic receptor and muscarinic receptor. So that's why acetylcholine can bind to any of them, because they have a structure similar to the muscarine and nicotine. Now the nicotinic receptor is an ion channel, and then the muscarinic receptor is a 7-pass receptor. Now, why different names? Yes, it is having different names like nicotinic and muscarinic. They're having different names because they are entirely different from each other. That's why I'm saying that here is an ion channel and here is a 7-pass receptor. Now let's go to where we can see the nicotinic receptors. Where we can see them. Now we can see nicotinic receptors on neuromuscular junction on NM. On NM. Okay, neuromuscular junctions. And we can see nicotine receptors also on nerves, like on ganglion. That's why I'm saying that for the preganglionic neuron of acetylcholine, at level of the ganglion, they release what? Acetylcholine and stimulate the NN, the nicotinic receptors. So nicotinic receptors are found at the level of the ganglion area on CNS. Okay. So on the neuromuscular junction, and then on the CNS, ganglion at the CNS. Now, muscarinic receptors, we have types. This becomes the type of nicotinic receptor, the types. We have NM and we have NN. Okay. NN are the ones at the ganglion, and NM are the ones on the neuromuscular junction. We have types of muscarinic receptors. We have M1, M2, M3, M4, M5. But go being so good, M1, M3, M5 are excitatory. That means that when acetylcholine binds to them, they becomes activated. But we have inhibitory ones. M2 and M4, the even numbers one, are inhibitory. The even numbers are inhibitory. So know the ones that are excitatory, that are stimulatory, M1, M3, M5. When I give acetylcholine to these receptors, they become stimulated. M2 and M4, when I give acetylcholine to them, they become inhibited. So note the difference. Now, acetylcholine can undergo configuration like nicotine and muscarine. I told you nicotine is having a particular structure that binds to its receptor. Muscarine is having a particular structure that binds to its receptor. But acetylcholine can undergo conformational change to fit any of them. So let's say this is a muscarinic receptor. That is how the ligand binding domain looks like. This is a nicotinic receptor. This is where the binding area looks like. Now, acetylcholine can change its form to this or can change its form to this and bind to any of them. That is why acetylcholine has got two receptors. That they have a nicotinic receptor and muscarinic receptor, because acetylcholine can change its form to suit any of them. So they can change to fit here or change to fit here. Now I have cholinergic drugs acting on the cholinergic receptor. We have direct acting and you have indirect acting. Drugs that directly go and bind to the cholinergic receptors are called direct acting. So drugs go there and bind to the receptor directly and cause stimulation. These are called direct acting drugs. We have indirect acting. They will not go to the receptors. They can even target enzymes like the acetylcholinesterase. It's not a receptor. And then bind to that receptor and bind to that enzyme and prevent the enzyme from eating up the endogenous acetylcholine. So those acetylcholine can bind to the receptor perfectly. So the point I want to emphasize here is that there are some drugs that directly go and bind to receptors. These are called the direct acting drugs. They go and bind to receptors directly and cause stimulation. Some of the drugs too do not go and bind to receptors. They target some enzyme somewhere, causing eating up the endogenous acetylcholine. So they go there and arrest the enzyme and allow the normal acetylcholine to go and bind. This becomes indirect acting drugs. Some direct acting drugs bind to nicotinic receptor and muscarinic, of course, we should know. that the drugs, some of them will go and bind to nicotinic receptors, others will go and bind to muscarinic receptors, because acetylcholine drugs can go any conformational chain to bind to any of them. Now here, let's look at this. So here is a neuron. It has released acetylcholine. Acetylcholine can go here and bind to this receptor, nicotinic receptor, or this acetylcholine can come and bind to this one, muscarinic receptor. That's what I'm saying, that nicotinic receptors are what? Ion channel. And then they'll ask muscarinic out the muscarinic receptors are what? 7 pass. Here for nicotinic, what they do is that when acetylcholine comes here and bind, it opens for ions to go inside and cause the stimulation. But for muscarinic receptor, when acetylcholine binds to this area, then it causes activation and starts. So here there are no ions that will come inside at the level of the muscarinic receptor. No ions will come inside. But for nicotinic, when it binds, acetylcholine binds, it opens a channel for ions to go inside. Those ions are going to cause the stimulation. But for muscarinic, when acetylcholine binds, then they activate this part and cause stimulation of the cell. Now, cholinergic activity, that is parasympathetic outflow. After dog chases you and you get to your room, you're going to rest and digest. This is going to be the parasympathetic. I told you that opesa what kind of padia. Now say about the friends saying about the sympathetic. I find say a dog is chasing you, and how ben should go in the body. You have a para and a parasympathetic up at them, who are them. Come on, chasing you, not by them, right? At that point, what are the changes that should go in the body? So after dog chases you and you get to your room, you're going to rest and digest. Until at that point, and there's release of what? Secretions. So that's where you're going to salivate, you're going to lacrimate, you're going to urinate, because you can't stand there and urinate, otherwise the dog will bite you. Into what them after now you're going to urinate. Do you understand? So diarrhea, that's defecation, bradycardia, after you understand? So you have bradycardia, your meiosis. Now you don't need much light to see far. So your people will constrict because of them. So your people will constrict. You don't need much light to see as far. You understand? You have meiosis. You have bronchospasm. Who you know? Anaka bronchus no. A B na airways to information from now to journey. Well, now you're not running anymore, so you need to close. Okay, you need to close. So you have bronchospasm. You understand? So you have excitation of CNS and skeletal muscles. They are going to be excited. And then you have lacrimation, salivation, and sweating. Now it becomes abnormal. Then the patient starts to pass out urine. These are abnormalities of the sympathetic nervous system. For secretions, they all belong to what? Parasympathetic. Until we will be a draw, parasympathomimetic. Parasympathomimetic are going to mimic the normal acetylcholine. That means that it is going to bring out this kind of secretions. Until we will be a draw, acetylcholine agonists. You should know that you're going to have secretions. Understand? So these are the things that happens in parasympathetic outflow. Dumbbells, and a very short form to remember is what? Dumbbell. That at the parasympathetic level, what is happening? Diarrhea, urination, meiosis. Okay, people constrict, bradycardia, and even accommodation. So your scleral muscles needs to constrict. Or let me say, sorry, to contract. So that to bring a lens for it so you can accommodate and see things and here. Because now you don't need to see things at far. So scleral muscle must contract and bring about accommodation. So all these things must go on whilst you're in the room. Now let's go to distribution of cholinergic receptors. Distribution of cholinergic receptors. Where can I see this kind of which types can I see it and where? You understand? Let's go to sphincter pupillae. Should be stimulated. Yes, your sphincter pupillae should be stimulated. And if it's stimulated, you should have what? M3. Because you say for the receptors, M1, M3, M5, they are stimulatory. M2 and M4, they are inhibitory. Maybe M2 will be a inhibitor. Maybe M4 will be a inhibitor. M1 will be an stimulator. M3 will be a stimulator. M5 will be a stimulator. So on the sphincter pupillae, we are all M3 receptors. So they're going to be stimulated. And once they're stimulated, it is going to cause meiosis. Your pupils are going to constrict because now you're not running anymore. so you don't need much light. Now your ciliary muscle should be stimulated. So M3 again. When they are stimulated, it brings the lens closer and you bring about accommodation because now you're inside. You need to see things at near, not at far, unlike we are running. So ciliary muscle should have what? M3 and to get stimulated. Lacrimal gland equals lacrimation, introduce secretions. So lacrimal glands must have M3. It needs to be stimulated so they have the secretions. Bronchus smooth muscles should be stimulated. So bronchus smooth muscles, bronchus, you understand? Now now we are inside. Anything will be I will say more close kakra. You understand? Until you say stimulating, by stimulating, we consume kakra. So your bronchus smooth muscles should be stimulated and you have what? M3 receptors. Heart should have M2 because now we've done we're using for fine. Now we don't need anymore. And you don't need your heart to beat faster. And you need M2 to inhibit that kind of speed. And the M2 is going to bring about low heartbeat. So bradycardia. Fast heartbeat is called tachycardia. But low is called bradycardia. Now GIT sphincter should be relaxed. M2. So your GIT should be relaxed. You don't need to be stimulated. If they are stimulated, you can't pass out urine. You can't pass out defecation. Okay. So it needs to be relaxed, and by relaxing, it opens up so that it can defecate. So M2 receptors are also found out there. Urinary bladder should contract to pass out urine. So M3 should be at the level of the urinary bladder because secretions. So you need to urinate. So you need to contract so that it can pass out urine. So they also have M3 receptors. Now, muscarinic drugs that binds to muscarinic receptors. Drugs that bind to, I told you there are drugs that can bind to nicotinic and there are drugs that can bind to muscarinic. Now let's look at drugs that binds directly to muscarinic drug receptors. Now we have two classes of drugs that binds to muscarinic receptors. Now the first one is cholinesterase. So cholinesterase, you have acetylcholine. Acetylcholine directly binds to muscarine because acetylcholine can change its conformation and bind to muscarinic receptors as well. So acetylcholine can bind to that. And we have synthetic preparation of choline esters, similar to acetylcholine. You have methacholine and carbachol and betanicol. These are all choline esters that are prepared, that are manufactured, and they all directly bind to muscarinic receptors. So choline esters bind to muscarinic receptors, and if not at all, know of acetylcholine, methacholine, carbachol, and betanicol. These drugs are choline esters, and they bind to muscarinic drugs. We also have another form of drugs that bind to muscarine. These are called alkaloids. And when you have alkaloid, it means that they are derived from plants. So we have some other drugs that are derived from plants, and I call them alkaloids. Okay. And we have a drug called muscarine. Muscarine too can bind to muscarinic receptor. You understand? And then pilocarpine can also bind to muscarinic receptor. So what am I talking here? There are a group of drugs that can bind to muscarinic receptors, and they are called muscarinic drugs. We have two classes of drugs, choline esters and alkaloids. The alkaloids are derived from plants, and we have muscarine and pilocarpine. These drugs can bind to muscarinic receptors. And for choline ester, you have acetylcholine, methacholine, carbachol, and betanicol. These drugs are choline esters. They all bind to muscarinic receptors as well. Now let's go to cholinergic receptor sites, or where we can see acetylcholine receptors or cholinergic receptors. Now on the CNS, I can see cholinergic receptors. On ganglion, okay, of parasympathetic or then sympathetic. That's why I'm saying that preganglion neuron release acetylcholine. They all go there and stimulate the nicotinic receptors. So they can all be seen at the ganglion of both para and sympa. Now, neuroeffector sites of post-ganglionic parafibers. So the parasympathetic also release acetylcholine. So they all also go and bind to acetylcholine or cholinergic receptors over there. And muscular junctions too, I'll see cholinergic receptors. So you're talking about where areas where we can see receptors. One, neuromuscular junctions. You can also see some in the CNS. You can also see some in the ganglion of both parasympathetic and the sympathetic nervous fibers. Are you okay? And neuroeffector site of post-ganglionic parasympathetic fiber. We can also see this kind of cholinergic receptors over there. Now let's go to indirect acting cholinergic drugs. I've talked about the direct acting. We are talking about pilocarpine and then carbachol, methanicol, they all go there and bind to the receptors directly and cause the action we need. Some of the drugs don't go and bind to receptors. They go and bind to enzyme so that it allows the normal acetylcholine to go there and fuse to the receptor. This becomes indirect acting cholinergic drugs. So we have them to have those who are reversible, those that can bind to the enzyme and leave the enzyme. Those that can bind to the enzyme and will never leave the enzyme. These are irreversible. So let's say, so we have the anticholinesterases that are reversible. Those drugs that binds to the enzyme and are reversible. You have a first drug called physostigmine. Physostigmine. And it's an alkaloid, meaning it is derived from where? Plants. And it's a tertiary amine. When you hear of tertiary amine, it means it is what? Lipid soluble. It can pass through the BBB. So this drug is an alkaloid, tertiary amine, lipid soluble. That means lipid soluble and can pass through the blood-brain barrier. Now, neostigmine is a quaternary amine. So it is not lipid soluble. And it is synthetic drug. It is not alkaloid. It is synthetic drug. Pyridostigmine, it is a synthetic drug. It is quaternary amine. So it cannot pass through the BBB. It is not lipid soluble. Endorphinium is a quaternary structure and it is a synthetic drug. So all these drugs here bind to the enzyme called acetylcholinesterase and inhibit it from it breaking down acetylcholine. But at a point in time, to leave the enzyme because it's reversible. Now, the first two drugs here, physostigmine, neostigmine, the mine, pyridostigmine, they are carbamates, meaning they're having a carbamate group attached to their structure, okay? And the endorphinium have an alcohol group attached to it. Now let's go to the irreversible anticholinesterase. Those irreversible ones that bind to the enzyme and never leaves the enzyme unless new ones are synthesized. This example of drug is called ecothiophate. This drug binds to the enzyme acetylcholinesterase and binds it forever until new enzyme is synthesized. Now, let's look at anticholinergic drugs. When I say anticholinergic, those drugs that act opposite to the secretion of acetylcholine to act on the receptors, so that I get more of acetylcholine. These drugs are going to decrease the production of what? Acetylcholine actions. I mean, acetylcholine actions. You understand? Now, you have cholinergic antagonist. You have cholinergic blocker, anticholinergic drugs. All these names are the same as anticholinergic drugs. They are the same names. Now, these drugs are classified according to the receptor they block. So we have some kind of drugs that bind to muscarinic, that are going to serve as antagonists. They're not going to do anything, but they will bind to the enzyme and they will not cause any action. They are inhibiting it. We also have drugs that go and bind to nicotine. So they are anti-nicotinic. They bind to nicotine receptor, but they do not cause any action. They are serving as antagonists. So for the muscarinic, we have non-selective muscarinic antagonists. They are non-selective. You know, I told you for muscarinic receptors, you have M1, M2, M10, all that. This atropine and scopolamine are non-selective. They bind to any receptor and inhibit it. Now, we have selective muscarinic antagonists. Some of the drugs that on the end say, may drug and make way, I am going to bind it only M1 muscarinic receptor. M2 have me inhibiting. So some drugs are very specific. They are very selective. I'm going to say I'm going to bind to one receptor per M1. That is what I'm designed to. An example is of the parasympathetic. They only bind to M1 selective receptors. And then M3, those who bind to only M3. Example here is delphina. They bind to only M3. So they are M3 selective. We also have, going to anti-nicotinic, we have those on the ganglion. So we have two types of the nicotine. You have the NN and the NM. This on the ganglion, and this is on muscle. We have two class of types of nicotinic receptors. Okay, so your nicotinic receptor and do ganglion, and as a receptor means when muscles. In 7% block on ganglion, I'll get a ganglion blocker. Ganglion blocker will block the nicotinic receptor on the ganglion. And a person blocking the nicotinic receptors on muscles, so I'll get neuromuscular blockers. And these blockers are going to block the nicotinic receptors on the muscles. So I'll end here. This is part one of the cholinergic pharmacology. We are not going to explain it very well, but we've summed everything up here. Thank you.